first slide. All right, good. Hi, everyone. Yes, I understand that this is the last talk, so I hope it's worth it. I want to thank Dr. Chen and the conference organizers for giving me the opportunity to talk. I've been listening in on to most of the talks yesterday and today, and all I can say is I really miss being there. It sounds like a great conference. Perhaps the next time I hear that it's going to be in Barcelona, so that's even better. My name is Vamsi Kodali. I am a data wrangler in the RefSeq group at NCBI. And I'm going to talk a little bit about rat genomics and some resources that are available to the scientific community today. All right. I'm going to first start with by introducing the NIH Comparative Genomics Resource, CGR for short. This is an NIH project that was established to facilitate reliable comparative genomics analysis for all eukaryotic organisms in collaboration with the genomics community. NCBI, as part of NLM, National Library of Medicine, is charged with leading the development of CGR and engaging genomics communities, with the vision to maximize the impact of eukaryotic research organisms and their genomic data to biomedical research. How do we get there? Well, CGR facilitates comparative genomics analysis through collaboration between the genomics community and NCBI. The image on the right here, represents how CGR works. In the middle is NCBI with its data, tools, and databases that can be used for comparative genomics. In the outer ring are community-provided resources that, with the community collaboration, can be brought into NCBI ecosystem through standardized interfaces illustrated by the arrows. What are the benefits for the scientific community? New and improved NCBI tools to screen for contaminating sequence and for annotating genomes will provide genomics community with a core foundation of uncontaminated and consistently annotated eukaryotic genomes for analysis. In short, high quality data. And some of the tools, particularly the FCS, foreign contamination screen, was already mentioned in a couple of previous talks. NCBI will also improve existing tools and develop new ones to improve and simplify comparative genomics analyses. These include new BLAST databases and comparative genome viewers. I'm going to talk about the comparative genome viewer briefly in this talk. NCBI will offer improved web and programmatic interfaces, one of which I'm going to talk about, to facilitate discovery, analysis, and the delivery of eukaryotic genomic-related content. So that's new and improved tools. And NCBI is developing additional tools to support emerging big data approaches to comparative genomics analysis, such as facilitating the creation of big data sets that are ready for machine learning and AI workloads and that are cloud-ready tools. This will meet new research needs and accommodate anticipated data growth. We've heard a lot about additional genome assemblies coming in for rats soon, so that's exciting, but that's a lot of data too, and we need to be ready for analysis of that. Put together, maximizing the impact of eukaryotic research organisms, we offer new possibilities for scientific advancement, particularly in the biomedical research. Why does what does all of this have to do with rat genomics? Well, as I was saying, CGR facilitates comparative genomics analyses through collaboration between genomics, community, and NCBI. And one such collaboration is between NCBI and RGD. NCBI and RGD have an ongoing collaboration that goes back over 20 years, way before my time. Over this time, NCBI and RGD have developed mechanisms that enable us to efficiently exchange large amounts of genomic data and utilize that towards improving nomenclature and gene annotations. RefSeq and RGD curators routinely consult with each other for cases that need special attention, such as gene merges and nomenclature updates. And RefSeq curators report genome problems that, for example, we have submitted about a couple of hundred issues that are related to RAT 7.2 genome. And all of these have bearing on the final annotation. Sometimes these conversations even include not just RGD, other nomenclature committees like a GNC, MGI for mouse, and this kind of concerted curation effort helps not just the annotation of rat genomes, but also other model and non-model organisms. Finally, we link out to each other. RGD pages link back to NCBI, and you can find links to RGD on NCBI pages. For example, this is an NCBI gene page, and RGD is listed as the primary source for nomenclature and provides the official symbol. And as part of the CGR project, NCBI gene now has descriptive information about genes that comes from the Alliance of Genomic Resources. For example, the summary section here includes a link to the pages of the Alliance genome. And in the case of genes that do not have a RefSeq curator provided summary, we use summary from the Alliance here. For example, this is from the Alliance of Genome Resources. Okay. So I'm going to switch gears a little bit and talk more specifically about rat genome at NCBI, MRAT BN 7.2. I don't have to introduce this assembly again to anybody, to this audience here. So I'm not going to even try, other than to just say that it was generated as back in 2020 as part of the Darwin Tree of Life and Vertebrate Genome Project, and was adopted by GRC, the Genome Reference Consortium, as the official rat reference genome. RefSeq group annotated this genome first in early 2021 using the NCBI Eukaryotic Genome Annotation Pipeline, EGAP for short. This annotation was a big improvement over the previous annotation release that was on RNR 6.0. In that, over 2,700 genes were added, and we realized a 50% reduction in the BUSCO duplicated gene score. I'll come back to this in a minute. RefSeq curators, in collaboration with RGD, have done extensive curation after this initial release. And just a few months ago, an updated annotation, RS 2023-06, was released. In addition to annotating the official 7.2 assembly, RefSeq used EGAP to annotate 3 other rat assemblies that were mentioned in a couple of the earlier talks as part of a pilot project. These pilot annotations are currently available in NCBI's genome browser called NCBI Genome Data Viewer, and or GDV for short. The default assembly is the RAT 7.2, and that's what's picked when you first go to GDB. But if you go to the drop-down menu, you'll see, along with the older assemblies, the three pilot assemblies for which we have annotation now. You can pick these assemblies and browse the annotation for these assemblies. In addition to this, the annotation products like GFF3, GTF files, transcript and protein FASTA files are all available to download from the FTP site as well. Okay. Before I touch upon a few examples that show improvements made to the annotations, I want to briefly describe the annotation pipeline. The RefSeq group at NCBI uses EGAP, the Eukaryotic Genome Annotation Pipeline, to annotate eukaryotic genomes. This is an evidence-based annotation pipeline where more and better quality input data produce complete and accurate annotations. So very briefly, the genome assemblies are masked and a bunch of sequences are aligned. Nucleotide sequences such as existing RefSeqs, cDNAs, ESDs from GenBank, same and cross-species proteins from RefSeq and GenBank, and a whole bunch of data from SRA, short read RNA-seq data, long read RNA-seq data, more recently CAGE data, are all aligned to the genome, and they form the evidence for the next step, which is the gene model prediction. Gene model prediction based on these alignments produces models that are optimized for full-length proteins and consistent with the available transcript evidence. These models are then categorized, named, assigned accessions, and then released to various NCBI resources for the scientific community. We've used this pipeline to annotate over 1,000 genomes so far, and more are in the queue. The URL shown here can be used to check which genomes have been annotated and when they have been annotated, which ones are in progress, and dig deeper into the annotation reports. Before I move on, I want to reiterate the importance of data. Better quality input data will produce better annotations, and the pipeline uses existing RefSeqs as inputs. So all of the manual curation work that happens even after an annotation run adds a lot of value to the subsequent runs and results in more complete and accurate data. With that in mind, let's compare three annotations. AR106 was first released. AR stands for annotation release. AR106 was released back in 2016 and was on the old 6.0 assembly. 108, released in early 2021, was on the first annotation on RAT 7.2. And more recently, we've released the RS-2023-06, which is an update to AR108. So if we look at just the total number of genes and pseudogenes, we see a significant increase in the number of non-coding genes in AR108 and the latest update. There's also a drop in protein coding genes going from 106 to 108. That's in part because 6.0 had some split genes that were annotated and had some false duplications that were annotated. And a further reduction is thanks to curation efforts, conversion to pseudogenes, link RNAs, etc. So now, if we look at the transcript, we don't see much of a difference unless we zoom into it a little bit. So if we look at it further, we see a dramatic reduction in the number of partial and gap-filled transcripts going from AR106 to 108. the number of transcripts with ab initio sequence, a purely computational predictions, was also reduced. There's a further reduction in the number of partials going from AR108 to RS-2023-06. There's 54% reduction. A good number of these partials have been either fixed or suppressed. This is entirely due to manual curation work by the RefSeq team. In collaboration with our RGD partners, over 7,000 gene symbols were updated in RS-2023-06. A majority of these were updates from something that's not quite as informative, like a look followed by a string of numbers, or an RGD followed by a string of numbers, to something that's a little bit more informative. And a good number of these for pseudogenes. Back in AR108, there were over 11,000 NCBI gene IDs that were not associated with an RGD identifier. But after the release of 108, the RefSeq team worked with folks at RGD, and now we have maybe about 300 gene IDs that without the RGD identifier association. Okay. Let's look at a few examples. We'll look at a few more of these screenshots. So let me take a few seconds to orient everyone. What you see here is a screenshot of NCBI's Genome Data Viewer. There are two genes tracks, one at the top and one here. The bottom genes track is from AR108, the older annotation, and the top one is the RS-2023-06. Let's start from the bottom. When we first annotated RAT 7.2, this gene was called look 100359968. Well, which, apart from being a mouthful, is not very informative. Then, more recently, this was updated. The transcript sequence here was extended at the 5' end to add a UTR, and the name, the gene symbol, was updated from the look to something a little bit more informative, PATE14. I don't know anything about PATE14, but it's definitely better than look, I would say. Another example is this one, where we actually have three genes here right next to each other. This one is going in the opposite direction. We're looking at the 5' end here. In AR108, it had an uninformative look number, but the more recent annotation has a more informative H4CL3 gene symbol. Same for example, in the middle gene. Previously, we had a name, but it was updated along with the transcript sequence, to which UTRs were added. And finally, on the right here, we have a pseudogene, which had a look number as the official symbol, but now has a much more useful pseudogene name, which also indicates the name of the gene for which it's a pseudogene. So that's kind of helpful. Another example, AHI1 gene. So this one is clearly in the previous annotation, which is shown here, was truncated at the 3' end. The protein was annotated without a stop codon. And this was manually curated after the release of AR108 to extend the 3' end based on transcript and RNA-seq evidence. And the 5' end was also extended to include an additional UTR-only exon. So this is all, again, driven by curation work. If we look at another example, clavicin 2 gene, this one was also severely truncated. It had an unsupported 3-terminus extension of an exon. It was missing 3 downstream exons, and many rodent ref-seqs, RNA-seq intron data, full-length transcript sequences showed that this gene encodes a longer protein. And that's why this particular transcript was completely suppressed, and a new one was created in its place, which better represents this gene. As you can imagine, there are a whole bunch of these, and I can go on and on. But since it's getting late for everyone, I'm going to stop and not go any further into the examples. But I will point out that if we do this using Busco to measure the annotation quality, we do see quite an improvement. In our case, instead of generating annotation de novo using something like Augustus or MetaUke, we run Busco in protein mode. We start with all proteins from the annotation, pick the longest isoform for each gene, and then run it against a set of Buscos. In this case, we run it against the glires, and as you can see, going from AR106 to 108, we see a good improvement in completeness score, and we see that further improve with the RS-2023-06. The high quality of ref-seq annotation in this case is, I would say, a direct result of all the manual curation work done by the ref-seq team of curators. I'm going to hurry up now. I'm going to switch gears one last time and briefly touch upon the many ways you can access all of the rich annotation data. The data generated by eGAP pipeline flows through a bunch of things. You see the BLAST databases that you can use for sequence searches. eGAP data flows into NCBI portals such as assembly, gene, nucleotide, protein, a bunch of files that are available in FTP, and data visualization tools like GDV, Genome Data Viewer, and CGV, Comparative Genomics Viewer. Just to briefly touch upon a couple of these, our latest redesigned way of accessing genomes, you can search by taxonomic groups, filter them easily, and you see a table with all the genomes, which you can either quickly download. You can either download just the table that you see here, or you can download all the genomes, transcript files, GFF3s, whatever you want, just with a single click. You can add or remove additional fields to this and use these tables for other analysis if you're interested. If you go to the redesigned NCBI genome page, you can see that we have the assembly information right at the top, followed by assembly statistics, sample details about samples that were used for the genome, and finally annotation details. I am running through this very quickly, but the idea here is that these redesigned web pages provide a lot of information that was scattered across multiple different resources within NCBI that's being brought into a single easy to use page. And you can even download the genome and genome data from this page just by clicking on a few buttons. Or if you are the kind of person who lives in the command line, you can use the datasets command line tool to download everything that you see on the genome pages from the command line. We have extensive documentation to help you get through this. We have installation instructions, how-to guides, and tutorials. And if you're an application developer, we have information about the API as well. So that's data sets. That's how you get the data. But if you're interested in visualization tools, Genome Data Viewer is NCBI's genome browser. You can search for specific organisms or use the tree here with the most frequently used organisms and browse through the genes. You can change assemblies. Earlier, I mentioned that RAT 7.2 is the default reference assembly, but we have annotations for a couple of other RAT assemblies as well. This is where you would pick that and search for your favorite gene and browse through them. This is the general interface of the genome data viewer browser. If you search for a specific gene, you'd see the familiar organization of multiple different tracks that allow you to browse through the genome data. And the tracks button here is a window to a treasure trove of data that's available at NCBI to add to this view. For example, all of the RNA-seq runs that were used for the annotation process, you can view their graphs, exon coverage graphs in this view by going through the tracks menu. And if you have your own data, you can always add them as user data from here. Finally, the latest tool, data visualization tool from NCBI, is the Comparative Genome Viewer. This tool allows you to compare two genomes. These comparisons are based on assembly alignments that were generated by NCBI, or in some cases, if they are available at UCSC, we import them. In the case of RAT, we have a bunch of assembly alignments, but considering that this is a relatively new tool, you may not find the alignments to your favorite organism. If that's the case, please write to us and we will do what we can to make those available to you. So let's say you select your assemblies, then you come to a screen that looks like this. A couple of previous presentations have shown screenshots like these. Essentially, these are two assemblies. The top here is the RAT 7.2, and the bottom is the mouse GRCM39 assembly. Both of them are aligned. The alignments are colored by their orientation. You can search for a gene.
Are you getting close?
Yes, I'm almost done. Sorry. Yes, I realize I'm running late. I'm almost done. So you can search for your favorite gene here and look at, zoom into that view and look at that particular gene as well. With that, I want to stop and just briefly mention what's coming up. The curation work by RefSeq curators continues, and we already have over 100 genes with improved nomenclature since the last release in June, and we're getting ready for the GRC RAT Assembly 8.0. In terms of EGAP annotation, the pipeline now can use CAGE and poly-A-seq data if that's available, and output RNA-seq exon coverage graphs in big format that will be available as an output on FTP. So this is something that you want to add to JBrowse or any other tool. That's something you can do. We are also going to generate gene expression counts for all the RNA-seq datasets that were used for annotation, and these will also be provided as part of the annotation output. With that, I want to thank a whole host of folks who are involved in it, particularly the RefSeq curators who've done quite a bit of work, and the leadership that has supported us throughout this process. And a big thanks to everybody who's still there. I know it's getting late. Thank you very much for your time and attention, and I'll take any questions you may have.